Hello, my name is Carlos Bloom, and today we're going to talk about the TARGT gene therapy platform for correction of anemia in end-stage renal disease. First, we need to define this pathology. When kidneys are diseased or damaged, they do not make enough EPO, which is erythropoietin. As a result, the bone marrow makes fewer red blood cells, causing anemia. When blood has fewer red blood cells, it deprives the body of the oxygen it needs. Other common causes of anemia in people with kidney disease include blood loss from hemodialysis and low levels of the following nutrients found in the blood, or in the food basically, which is iron, vitamin B12, and folic acid. Now I'm ready to further explain this topic about the anemia in the chronic kidney disease.
Anemia in chronic kidney disease is a potentially serious medical condition in which patients have low levels of hemoglobin, a protein that carries oxygen to cells throughout the body. Millions of CKD patients in China and the US experience this condition. For severe CKD patients, their anemia can increase the risk of heart disease, stroke, and hospitalization. We asked 1,000 CKD patients about their perceptions and experiences of anemia in CKD. Patients in our survey found that anemia in CKD has a big impact on daily life for patients, including feelings of sadness. In both the US and China, many of the patients surveyed are also unaware of what common symptoms to look out for, including headaches and difficulty breathing. Discussing and tracking hemoglobin levels is an important aspect of disease management. But our survey found that many patients need more support to understand their hemoglobin levels, and patients would like more information. So, what if we started having more conversations about the potentially serious risks of anemia in CKD? Those patients we surveyed who felt more confident were more likely to be informed by their healthcare professional of the symptoms, impact on daily life, and severity and impact of anemia in CKD if left untreated. CKD patients should feel supported to take a more active role in managing their anemia. At AstraZeneca, we put patients first. We are committed to listening to the patient voice to improve outcomes.
Now we need to talk about the application in medicine. The transduced autologous restorative gene therapy, or TARGT, is a new autologous protein delivery platform designed to provide sustained suppression of proteins such as the recombinant human erythropoietin, which uses a small autologous dermal explant or explants in which sterile biopsies samples of the given patient dermis are transduced ex vivo with the use of a helper-dependent adenovirus system that's engineered to carry this gene. Now, to further explain the topic about the gene therapy, here we have a video.
Many human diseases are caused by defective genes and improper protein functioning. Genes within our DNA provide the blueprint for the production of proteins. Specific gene defects or mutations can result in reduced or inability to produce a functional protein or production of a protein with altered function. Gene transfer using adeno-associated virus, or AAV, vectors can target specific cells to correct disease-causing improper protein function. Once inside the target cell, therapeutic genes within the AAV can correct defective or missing protein function. To achieve this, AAV genetic material is replaced with a therapeutic gene to restore natural function of the disrupted proteins. A protein shell, called a capsid, encases the genetic material and helps target delivery to certain cells. Once within the target cells, the capsid is shed inside the nucleus to release its vector genome. The vector genome does not integrate into the nuclear DNA, but instead forms an episome. Episomes are not passed on to future generations of cells, thereby ensuring that the effect of the gene transfer is not passed on. Using the body's natural machinery, this allows stable and long-term expression of the therapeutic molecule. AAV-based vector delivery systems can be administered by intravenous drip or direct injection to target tissues. AAVs are ideal vectors for gene transfer, as they are non-pathogenic and capable of transducing both dividing and quiescent cells. The different components of AAV-based gene transfer can be switched to address different therapeutic targets and diseases. And several studies and clinical trials have indicated that there is activity in different disease models. Gene transfer offers the potential of long-term and potentially curative benefit to patients with genetic or acquired diseases by correcting defective or missing protein function through a single administration. uniQure is committed to developing innovative and cost-effective single-treatment gene therapies with potentially curative results.
Now, we need to talk about the pros and cons of this type of treatment. Now, the pros is that there were no serious adverse events related to the TARGT EPO, which means that the treatment using the EPO genes had no adverse or important adverse events. And also, there's no erythropoietin antibodies, therefore not reducing the erythropoietin created by this treatment. Now, to refer to the cons, is that this does not allow us to assess the risks or the benefits of the target EPO as compared with the administration of recombinant erythropoietin. Therefore, it requires a prospective randomized trial, which will give us a better understanding of what's happening. The issue applied in Ecuador. About the 0.066% of the population have this type of renal disease. And this percentage increased by 10% per year. Therefore, by now, with the actualized population, it will be about 50 million people with this disease in Latin America. In Ecuador specifically, we're talking about 1 million of people that have this type of disease. Therefore, this type of treatment will be very helpful to a lot of people here in Ecuador. Talking about the conclusions. We need to conclude that the sustained levels of erythropoietin and hemoglobin after TARGT EPO implantations were achieved with the maximum plasma level of erythropoietin that was approximately 1/100th of the maximum level measured in the patients when they received a recombinant human erythropoietin by injection. The early study that does not allow to assess the risks or benefits of the TARGT EPO as compared to the administration of recombinant erythropoietin injections. Therefore, as it was mentioned before, more studies should be made. Thank you.